Next up is Ginger, and I hope I'm saying this right, Ginger Sung. Oh, sorry.
Everybody gets that one wrong.
Okay, so. And we will make all these slides available with the presenter's permission on citsibio.org, is that right? So you can find them there. Thank you.
Alright, hi, I'm Ginger. I'm from Anderson's lab at the Swiss Research Institute, and I work on Mark2Cure. And Mark2Cure is essentially an attempt to use citizen science to make this unwieldy knowledge dump known as biomedical literature more usable. And to clarify this, let me talk briefly about the process of biomedical discovery. And so traditionally, researchers read papers to find out what's known and what's not known about a subject. And then they synthesize what they've learned from those papers to generate testable ideas. They test these ideas, they generate data, they organize and structure that data into, try that. They generate and test those, they structure and organize that data into information, they integrate that information into knowledge, and then they take that knowledge and they dump it into a paper and disseminate it in hopes that you will eventually, or the relevant people who need it will eventually find their work and use it to advance the field. And the problem with that is that there are so many papers published. I think we're at about 26 million citations in PubMed and growing about 1 million articles per, or a little bit more than 1 million articles per year. And if you were to try and read all that, it's impossible. And so essentially, biomedical researchers are generating their hypotheses on narrower and narrower slices of the entirety of biomedical knowledge. And the problem with that is that in biomedical research, everything is related. So if you are able, and props to you if you can, keep up with the literature in a very narrow slice of your discipline, it could be that the clue you're looking for your big breakthrough is actually right outside of that narrow slice. So we'd like to make more of that the knowledge that's buried in the biomedical literature usable. And a lot of people have this goal. So there are a lot of efforts to manually curate and mine information from biomedical literature. For example, Reactome does some really expert curation efforts there. There's also a lot of natural language processing tool development that's going on. And these natural language processing tools do have an important role to play in biomedical literature mining and discovery. And one of these tools is the PubTator suite. They have a nice algorithm for identifying gene mentions, chemical mentions, disease mentions in biomedical text. And this tool is not a substitute for manual curation. It's used to assist manual curation. So the bottleneck is still finding those manual curators. And the question is, can citizen scientists help with that? So, short answer, we ran a beta study to look at whether or not citizen scientists could help with that. And we essentially trained citizen scientists to identify disease mentions in an already expertly annotated disease corpus. And what we found was that citizen scientists could do this task, and they could do it really well. In aggregate, their results were on par with that of an expert annotator. And so, since they can do this, and they can do it well, we've expanded Mark2Cure to encompass not just disease mentions, but also gene mentions, treatment mentions, as well as identifying the relationship between these different entities, because this is really where machines are not that great at doing this task. So identifying the relationship between concepts is still computationally very, very tricky. And although we still have a long way to go in terms of the number of abstracts we would like our citizen scientists to do, we feel like Mark2Cure is really shaping up in becoming a tool in the biomedical big data toolbox. And our curators are unexpectedly becoming quality checkers for various biomedical data resources that experts use. So, for example, we had several users who identified duplicate entries in our system, which we sourced to PubMed, and then when we contacted PubMed, they removed the duplicate entry. And we've had curators who noticed that certain drugs are missing from biomedical resources such as DrugBank, and so we contacted DrugBank about that, and within a week they added a sub-entry for that drug. And we've had users find kind of where PubTator is struggling. So we pre-populate our abstracts with annotations from PubTator to make it easier for our users. And PubTator identified WD in these abstracts as Wilson's disease, and our users threw those out. And when we looked as to why, it's because in these abstracts, WD actually represents tryptophan aspartic acid repeats. And if we take the identifier Wilson's disease that PubTator associated with the string WD and looked into PubTator, it gives like about 9,000 or greater than 9,000 abstracts that PubTator annotated with this identifier. And at a cursory glance, because I wasn't able to look at all 9,000 abstracts, but just looking cursorily at the first 100 or so, it looked like unless the text specifically says Wilson's disease, the WD probably doesn't refer to Wilson's disease. So there are areas where PubTator can obviously improve, and we think that citizen science can help with that. And so anecdotally, we've seen that even though we design Mark2Cure in hopes of pulling facts and data out of the biomedical literature so that we can do hypothesis generation with bigger portions, more comprehensive portions of the literature, we're finding that users are using citizen science in biomedical literature to help improve existing individual data resources such as DrugBank. They've provided feedback which helped improve entries in PubMed, and we're also seeing potential improvements for PubTator. So there's some anecdotal evidence for this, and we've only done about 2,000 or so abstracts. We think if we expand the amount that's actually done, a lot more interesting improvements will come out of this. And we're not the only ones in this space now, so we're not the only ones to acknowledge that biomedical literature is important and a very good entry point for citizen science. So last May, Cochrane Institute started a citizen science project called Cochrane Proud, and they essentially use citizen science to screen for randomized controlled trials. And they hope to use the screening process in order to find health evidence. So we think it's going to be a very useful entry point for citizen science into biomedical research. And with that, we'd like to acknowledge everybody who helped in this project, and especially the citizen scientists.